This Week in Virology, the podcast about viruses, the kind that make you sick. This Week in Virology. This is a special episode recorded on August 25th, 2015. Hi, everybody. I'm Vincent Racaniello, and you're listening to TWiV, the podcast all about viruses. Today I'm coming to you from Princeton University, which is in Princeton, New Jersey. I've spent a lot of time here because this is where we work on our textbook, Principles of Virology. In fact, I'm in the Lewis Thomas building. I'm on the second floor, and this is actually not far from where we had our textbook meeting for the past 2 years, almost once a month. But today I'm not writing a textbook. I am visiting a scientist here in the Department of Molecular Biology, where she is a professor, Ileana Cristea. Welcome to TWiV.
Hi, Vincent. Thank you for having me.
Did I pronounce your name correctly?
You did.
Ileana Cristea.
I will take that.
Yeah, okay. I know I'm sensitive to name pronunciation because people pronounce my name incorrectly often.
Oh, no.
Can you pronounce my name?
Racaniello.
Very perfect.
Yes,
perfect.
sure.
You must have been born in Europe somewhere.
Yes, indeed I was. Indeed I was.
So I would like to today
Latin-based.
talk a little bit about your science, a couple of different aspects. But before we do that, I want to start by figuring out where you're from and where you were educated and how you got here. So you're born in Europe. Where were you born?
I was born in Romania, in Bucharest, so I apologize in advance for this accent.
It's a great accent. I love it.
Sure. Okay. And so I was born there. I started my education there. Mm-hmm. I loved biology and chemistry from a very young age and started at medical school. Figured out very quickly that I would love to figure out actually how things work and the science behind the problems, and I all the time ask what's the cause of this disease. And so I started in the same time a chemistry undergraduate degree, and I moved to England after that. I continued my undergraduate degree there at UMIS, so it's University of Manchester Institute of Science and Technology, that now became University of Manchester. And I continued there with my master and PhD before moving to United States.
So this was in chemistry, your PhD?
That was in chemistry, yes. So my master was in medicinal chemistry and my PhD was in chemistry. And also that's where I met Simon Gaskell, who introduced me, a professor at UMIS at the time, and who introduced me to mass spectrometry and proteomics.
So that was your intro to this interesting
Yes,
analysis.
it was during my undergraduate degree.
Was there any science in your family?
Yes, my father, Paul Cristea, was a fantastic scientist and a member of the National Academy of Sciences in Romania, and he was in physics. But he definitely loved multidisciplinarity, and he did digital signal processing and tried to translate DNA, signals into to try to understand information and try to understand the evolution of life as well. So he was very passionate about science.
So how far in your career did he see you? Up to what point?
He saw me until I actually joined Princeton.
Wow.
Yes.
So he must have been really proud of you, right?
He was a tough guy. If he was proud, he would not necessarily let you know. He would let his friends know.
And then you'd find out?
I would find out from his friends.
Okay,
yes.
But he must have loved what you were doing, right?
Yes. Well, he hoped very much so that I would go into computer science and engineering. So for a while, it was a bit of a disappointment until I actually got to Princeton, and he thought, okay, I must be doing something right. But yes, really remarkable scientist.
How about your mom? Was she involved in science?
My mother, she was in journalism and was involved in news and in journalism, in newspaper. She had quite a high position in what would be the equivalent of New York Times here, so the pretty prominent journal, Evenimentul Zilei, in Bucharest, in Romania. So you can imagine that was a fun time going through communism and through some tough system to write about news.
So she was a news writer, not a specialist of any
She
kind?
was. She actually had a degree, a university degree in literature.
Okay.
So, but in order for both of my parents to be able to stay in Bucharest, she changed careers and did that.
Okay.
And my grandfather was involved also in chemistry.
Oh, so you have significant science in your life.
Yes.
In your family, yeah, great. All right, so then PhD in Manchester.
Yes.
And what was next?
With Simon Gaskell. And as I said, Simon is a really tremendous pioneer in mass spectrometry, and it was great to be introduced to really the basics of mass spectrometry in that lab. He worked on discovering how peptides fragment and how they generate ions and how we are able to detect them, and he would build instruments as well. So I think he made mass spectrometry an easy thing rather than a black box. And after Manchester, I moved to United States, to Rockefeller University, where I did my postdoctoral research with Brian Chait. And Brian really loves mass spectrometry, but loves also biology, and that's something that we had in common. And given my background in medical science, I always looked for opportunities to bridge these two fields, proteomics and biology, and I was able to do that very well during my PhD and my postdoc.
So did you pick the postdoc lab based on the fact that you wanted to use mass spec to try and solve biological problems?
Yes, exactly that. And when I joined the lab, I actually joined for a project to work on in neuroscience project with Ned Hines to try to understand what makes synapse excitatory versus inhibitory, and what is really the composition, that signature composition. And that was a pretty long-term project and quite a challenging one. But that allowed me also in the same time to start my own projects in parallel with something that I was really keen to start to do, and that was to bridge virology with proteomics.
So your interest in viruses began at Rock, you think?
My interest in viruses started earlier on, even during my PhD. My PhD was funded by GlaxoSmithKline, and it was based on toxicology and also on viral infectious diseases, so I got quite interested at that time. But where I started to implement this more and more was really at the Rock, as you said. And I spent quite a bit of time there in the laboratory of Charlie Rice, who allowed me to go there and use all his hoods and grow the cells and infect them. And
So I know, I call it the Rock because I have a little history. My postdoc advisor, of course, was president for a while, David Baltimore. He got his PhD there. I don't know if you knew that.
Yes.
Lots of famous virologists actually got their PhD at Rockefeller. Bob Krug.
Right.
Dick Compans, and there are others, but I'm not remembering them. Anyway, I was a graduate student in New York, so I often went to Rockefeller for various talks or meetings. And then when I came back to New York to work at Columbia, my wife was a postdoc at Rockefeller. I think this was from 89 to 90-something. And we lived in Manhattan, and on Fridays I used to go to the faculty club to drink with all the
famous faculty club.
Yeah, the famous, and they would have pictures of their previous students, and David Baltimore was there as a young person, and many other people. So it was a very interesting place. Got a lot of history.
A lot of history, and it's a very inspiring place to do science.
And of course, on the East River there, it's beautiful location.
Beautiful views. Yeah.
So, did you come to Princeton out of Rockefeller?
I came directly from Rockefeller. After my postdoc, I came here to the Department of Molecular Biology as an assistant professor. And it's interesting when you're interested so much in two fields of studies, because there are either virology labs that are wonderful, pure virology labs, or there are these mass spectrometry labs that can be either small in size and focused on technology developments, or they can be really large and based on many, many collaborations, and they are wonderful labs. But I wanted to bridge this properly and not have to rely only on a collaborator in order to work on a biological question, or have to bother Charlie Rice or somebody to be able to do these experiments. And that was my dream when coming to Princeton, that I wanted a lab where the lab will truly be integrating these two fields of study properly, and that everyone in the lab would be trained in proteomics and virology, and we wouldn't have just the guy working on the instrument and fixing this and the guy doing the virology. We would have them, really everyone would have to learn both skills.
And that was probably an attraction for Princeton, because they probably wanted someone who could do both, right?
I hope so.
They didn't want a person to be a service for everybody else and do their mass spec. They wanted someone to do research. And of course, having so many great virologists here was also a plus, right?
I have tremendous colleagues, and that was definitely one of the main factors for me joining here, this department. We have, of course, Tom Schenk, who has become a long-standing collaborator and friend of mine, and it's been an absolutely terrific collaboration. Lynn Enquist, who can be blamed for hiring me here.
He was the chair.
He was the head of the department
when
he hired me, and has been so supportive of my career and my lab and everyone in my lab. And Jane Flint, of course. And just recently, just a few years ago, we hired somebody else from Rockefeller University, from Charlie Rice's lab, Alex Ploss. So I'm glad that this tradition continues here, and that we continue to have strong virology.
We had also made Alex an offer, so he decided to come to you instead of us.
Very wise guy.
A very smart decision, because if I were starting my career, I would try to come to Princeton. I think it's an example of how you can have a great undergraduate institution doing research.
Yes,
really supportive.
All of our undergraduates, as you say, have to do research in the lab in order to get a senior thesis, a senior research thesis, and to graduate.
So when you applied here, what was, usually you have to write something about what you're going to do, right?
Yes.
So in broad terms, what was that about?
So it was about understanding the roles of chromatin remodeling complexes during viral infection. It had to do with this observation that I had from my studies of virus-host protein interactions, where I looked at quite a few different viruses. My first work was done on Symbis, a small positive strand RNA virus, right? So I worked on Symbis, but then I worked on HIV, and then I worked on several herpes viruses, on human cytomegalovirus. And it seems that all these different viruses were somehow modulating these important enzymes in our cells, chromatin remodeling enzymes that regulate the epigenetic landscape of a cell. So I wondered if that's a really elegant way through which viruses can modulate viral gene expression, but also host gene expression, and create an environment that is beneficial for viral replication and spread.
And that's a problem that's amenable to mass spectrometry, right?
Yes.
So let's talk a little bit about that. When I was a student, which was in the late 70s, nobody did mass spec in labs. There were a few here and there because they were so expensive, and no one could just have them as part of a lab. But that's changed. And what happened?
Although not the expensive part. Unfortunately, not yet.
Why is it that there are more mass spec devices than there were when I was a student?
So it started kind of in probably mid-90s, when it started to be really popular, and in terms of really the integration of mass spectrometry within biological projects. And that's, I think, what created this boom. When we were finally able to to ionize a molecule, a biomolecule. And that was introduced by this, the electrospray ionization and MALDI ionization. So either ionizing a sample in a solution or ionize it in sort of a crystal form with a laser that generates these ions. So before this, it was really hard to look at molecules, at biomolecules, like a protein or a lipid, and I think that's what triggered this expansion and this interest in generating and designing better and better mass spectrometers that have higher sensitivity, higher accuracy. It's when I started in this, analyzing something at the picomole level was considered extraordinary. These days, it's analyzing something at atomole level is considered routine, right? So
before this, before these techniques, it was only possible to look at inorganic compounds?
Yes. Most applications were more towards chemistry projects.
So let's talk a little bit about how it works, because I
Yes.
think our listeners would like to know. It's a big machine. Maybe you can show me it later. And so it's big, and you need someone who knows how to run it. But what does it actually do?
So every mass spectrometer, it's actually fairly simple in concept. So it has an ionization part where you're introducing a sample and you're generating these ions. Without ionizing this, you cannot detect your samples. Okay. And then it has a mass analyzer, and this is when you can have all these different instruments, right? For example, different types of mass analyzer. You can either, once you generated the ions, you can either measure the time that it takes for those ions to reach the detector. So that's called something like time of flight.
That's where MALDI-TOF comes from, right?
Yes, exactly.
Okay.
And of course, the better that resolution is, the better you measure the time of flight. So then you can have some sort of mirrors, what we call reflectors, to kind of expand the time of flight of the ions. You can have another type of mass analyzer that is called an ion trap. And it does exactly what it says. It traps the ions until you have enough to send them to the detector. And this is something that it's very sensitive. This is what they send to Mars to find out what's the composition there, because it's so sensitive. You accumulate ions and you send them to the detector. In terms of the biological applications, this is what's used now for targeted mass spectrometry. If you want to find out in a really complex system, do I have that particular protein there? Or at what amount is that protein present? Or is there a certain post-translational modification on my protein? You can do this in a targeted way and really effective. And of course, probably the most sensitive and the one that most accurate instrument, Orbitrap, it's probably the one that is most spread, invented by Alexander Makarov at Thermo. And Orbitrap, again, the ions orbit around this inner electrode, and you measure the frequency of orbiting. So you have this ionization device, you have mass analyzer that can be any of these combinations that I told you about, and quite a few others. Usually an instrument won't have only one mass analyzer, they will have several mass analyzers so that you can analyze your sample in different ways. And then there will be the detector at the end that detects these ions. And then, of course, really the most time-consuming part is data analysis, or the bioinformatics that for a while lagged behind, but now it's really catching up, and there are better and better programs and software to deal with the large amount of data that is being generated.
So besides being a chemist, you also had to become a computer scientist.
Aren't we all?
Yeah, to a certain
These
extent,
days we have to, right?
I think some of us are more than others.
Yes.
Right? One of the applications for MALDI-TOF that I know of is identifying bacteria in clinical labs.
Yes.
Is that correct?
Right, it's correct.
So how would they do that? They would take a clinical specimen and put it right into the machine, or do you have to process it in some way?
It's very simple. You can take a clinical specimen. You can you can do this in different ways. You can either place it straight into a matrix solution that absorbs at the wavelength of the laser and generates these ions. And if you have already signature ions that are present and they are formed even without digesting your sample, you can do just that. Or usually you would digest with an enzyme, generate all these pieces, peptides, and then analyze that. And that's probably has a bit higher accuracy.
Okay. So basically it tells you proteins that are present, and
yes.
then you use
and you can look at yes, it tells you the proteins, and you will know if those correspond to a certain bacteria, a certain species, or a certain virus, and you look for these signature molecules.
So will it tell you the composition of the protein fragments? Is that
yes.
And then
yes.
you use software to figure out what the sequence would be?
Yes, right.
By matching it to something that's known.
Right.
So you can't do unknown things though, right?
You can do unknown things, but then you're relying on a really high quality spectrum or high quality fragmentation of that peptide or that protein. And actually, when I started this, the databases were really incomplete during my PhD work and so on. So I would have to manually sequence most spectrum and find out what the amino acid sequence is, which I absolutely loved. It was like a little detective story. And even now, I sometimes ask my students to do this. And one of the professors from University of North Carolina, Frank Conlon, did a sabbatical in my lab. And I first asked him to sequence this manually. And he was, oh, no, and now I have to do this for every single peptide. And I told him, no, now you can do it automatically, but you know how it works.
So there is a database, you just made reference to this,
yes,
of data that you can compare your results to, and people contribute to this? Tell us a little bit
Yes, so
about
there
that.
are different types of databases, either on theoretical proteins, what the sequence should be, and therefore if you know that you're using an enzyme, let's say trypsin, that cleaves after certain amino acids, in this case after arginine and after lysine, you already can generate a theoretical digest with the peptides for any possible protein. And then based on that, you can also generate theoretical spectra, right? Where if you would have that peptide, what fragments would it give you once it's fragmented? All these, what we call BNYs, what are the fragments between two amino acids that can be formed in a mass spectrometer? So there are theoretical spectra, and then there are also experimental data sets. So there are databases of accumulated spectra on different instruments that you can just match your sample and that match your spectra with, and that's sometimes even more accurate because then you take into account the fact that a peptide can ionize with much higher efficiency than another peptide. So therefore, or a fragment can ionize better. So these won't be equal ions or equal peaks, right? They will have different intensities.
So are these databases available publicly?
They are available publicly.
So everyone who does mass spec shares them?
Yes.
Even forensic laboratories, for
Even
example?
forensic laboratories. When I was in England, we actually had quite a few forensic applications, and few of my colleagues were funded by forensic grants.
Okay.
Yeah.
So when you came here, part of the deal was to get you a machine, right?
Yes. So part of my startup was, thank goodness, to get one instrument, and then from another grant, I purchased another instrument.
So you have two now? I
have two, and I have a really very good ongoing collaboration with Thermo Scientific, and they have been great at interacting with.
So what do you call yourself? Are you a virologist or a chemist or what? Do you fit into any name?
I am an aspiring virologist. I'm a student of virology. I keep on learning, and a student of proteomics probably.
Okay.
I find it hard to define myself, and that's a problem with when I go to a proteomics conference or a mass spectrometry conference. I may be seen as the virologist or someone who brings that outside technique. In the same time, and when I go to a virology conference, I probably am the proteomics guy. But I think it's important to actually try to learn both of these fields properly.
Well, you're using proteomics to study viruses, basically, right? You don't study bacteria or fungi or protozoa, it's viruses. It's not, you don't study human problems. So I would say you're a virologist. And in fact, we should say I had you as one of my morning speakers at the ASV meeting this summer, which is a virology meeting. And
that's a wonderful conference, absolutely wonderful. I came back so inspired and immediately told my students that they have to go there and attend this conference. It's really a tremendous...
I'm very happy you came, but I look at you as a virologist having, we have discussed your work previously on TWiV, and I view you as a unique person because you know how to do the mass spec and analyze it and how to figure out how to apply it to virological problems. Most virologists can't do that.
Yes.
And they have to find people like you to collaborate with. And
I think that's absolutely an advantage to have such a technology in your own lab, because many times you have this problem and you have a question in mind and there is no method out there to address it. But having this in your hand gives you that flexibility to be able to think about how you would design a workflow or a new method to address an issue.
So here's an idea I had. I wanted to do this years ago, but we don't have any mass spec that
Yes.
we can do this. I work on picornaviruses, right? And the
Right.
assumption has always been they are capsid proteins and RNA, and that's it. But maybe there are other proteins in there that are
Right.
important. And I said, why don't we take purified virus and do mass spec? Was that a viable experiment which would tell you something, or is it too prone to contamination?
So that's definitely a good experiment, and I think that that's one of the applications that mass spectrometry has been used for in virology. But I think then you have to think about how pure your virions are, and are they all homogeneous, right? Or is there a heterogeneous population? How mature?
Right.
Because mass spectrometry will not lie, will tell you everything that is there, but will not tell you if something is just attached to a virion from the outside or still from the cell. So for that, usually people have to include more and more controls and kind of design their experiment to take that into account. Maybe have some digestion.
Yeah, we could digest
yes.
with an enzyme because
Exactly.
the capsid is relatively resistant to trypsin.
Yes, right.
And can get rid of
You
other
can get
things,
rid
right?
and see what still remains. And
So we just mentioned the proteome, which is, of course, all the proteins in a system, right?
yes.
Just like the genome is all the
Just
nucleic
that-
acids.
Yes, just that the proteome is so dynamic that I think it's sometimes hard to define, because the proteome may depend, even our own proteomes won't be the same in the morning as in the afternoon
Sure.
or in the evening. So the same with a cell that we grow. I think it's really not a static, well-defined thing. And in most of our experiments, we always have an average of proteomes in reality, right, from all the cells that we are looking at. And a protein, of course, most times will have many different functions and will be involved in many different protein complexes, or will travel in different subcellular compartments.
Sure.
And what you'll analyze within that proteome is a mixture of all that.
Of course, of course. And of course, most of your work is done in cell culture, right?
Yes, most of the work is
Interesting thing would be to look in animals and
Yes.
say, what does a virus do in the CNS and
Yes.
whatever tissue, what does it do to the proteome?
And
what does that
definitely,
mean, right?
and definitely proteomics is amenable to that, and there are many people who are doing not only in virology but in many
Sure.
other fields. We are also using some animal models, and we probably will talk later about some of that.
So can you put a whole mouse into a mass spec? No, you can't do that.
No, but at one point I remember we were showing how easy it is to do these analysis, and brought a kiwi and a syringe and just extracted some kiwi
really?
and put it on the MALDI top, as you said, and say, okay, you see, you can prove to the
Cool.
students that in 5 minutes or 10 minutes you have something.
Now, you have another ohm that I have heard you use, and it's in your papers. It's called the crapome.
Oh, the crapome, yes.
I know what crap is. It's the stuff we don't want. And so what is a crapome to you?
It's such an eloquent
It is.
terminology.
Is it actually an acronym for something?
So yes, probably the contaminant repository. By the time I would tell you this, I would forget how it started. But so in short terms, it's a repository for non-specific associations. So whenever you're doing an experiment where you're interested in finding out information about protein interactions in a cell or in a tissue, in a mouse, in any sort of system.
Could it be a virus too?
A virus, you want to figure out what are the likely interactions that are real and which ones are likely to be non-specific.
Okay.
And given the fact that many systems out there use all these tagging approaches where your protein is tagged with, let's say, a green fluorescent protein or a protein A or a MYC, or that can influence those non-specific associations. So that crapome is a large repository where 12 labs and probably more now have contributed all these different controls that we have done for many, many years in many different cell types and in many different, with many different systems, lentiviruses, retrovirus transductions, or just wild type cells, so that people can use that, and it's made available to the scientific community for everyone to use. Alexei Nesvinsky and Ankolod Gingras were the ones who really thought of this, and Alexei Nesvinsky is a professor at University of Michigan, a bioinformatician, so he's the guy who put all that database together to make it accessible.
So basically you get your results and you can run them against the crapome
Yes.
to subtract what's probably not significant.
Right, or at least it's going to give you a feel for if this is a protein of interest, or from your proteins of interest, is it likely that it appears through a non-specific association? So it's going to tell you if suddenly your protein of interest is in 80% of interactions out there for that particular system or in that cell, you may want to know about that.
So has any entry in the crapome been subsequently shown to be real? Because you can imagine that that might happen.
Oh, and this is really an important point. I would say that that's just a reference for you to know about, so that you know.
Right.
But it's really challenging, and the ones that are most challenging to prove as a real interaction are the ones with abundant proteins. Let's say actin, right?
Right.
Or vimentin. Most of us have those proteins. Most of us have them as non-specific associations, and nevertheless they will form a lot of real interactions.
Okay.
So for those sort of things, you can use other methodology to really determine if that's a real interaction. Something IDERT, for example, that uses metabolic labeling in cells with stable isotopes. It's a really good method to look at this sort of more finer information about specificity of interactions.
So what you put into a mass spec machine can have radioisotope labeling as well to help you narrow down the possibilities?
And to help you with the quantification, right? The relative quantification between different samples, let's say different stages of viral infection. That's really great approach.
So maybe now we can talk about some of your experimental work and tell our listeners how mass spec fits into it.
Yes.
Right? Because I would guess it touches almost everything you do in the lab, right?
Right.
In one way or another.
Yes.
And so one area that I'm pretty interested in, we've talked about on TWiV, is DNA sensing, sensing of DNA viruses. And I was interested in this because we work on RNA virus sensing. And I always heard talks by people who worked on the DNA side, and they're always finding sensors in the cytoplasm.
Right.
And this never made sense to me because most DNA viruses, with the exception of the pox viruses, they go into the nucleus to replicate. So they enter the cells, and the capsid goes to the nucleus, and the DNA goes in. So why would DNA sensors in the cytoplasm didn't make sense? And we've mentioned this on TWiV. And then Lynn Enquist wrote us an email and said, you don't know the work of my colleague, Ileana, who's found nuclear sensors. Because I think we said there has to be nuclear sensors. So let's talk about that story, nuclear sensors for viruses. How did you get so interested? Because you told me when you came here you're looking at chromatin remodeling. Right. Right. So
it all started from this interest in understanding virus-host protein interactions during the progression of an infection. So if we can infect cells and use full-length replication-competent viruses that are tagged with fluorescently labeled tags, like green fluorescent proteins, and those are present at different locations, so different viral proteins would be labeled as they are expressed in the cells. If we can really follow the life cycle of a virus in cell, but then also isolate those viral proteins at different time points, we can get information about what the viral proteins actually target. What do they need to inhibit? What do they need to target in the cells as the infection progresses?
Okay, let me interrupt and just ask you a question. Let's say you have a viral protein that's labeled with GFP.
Yes.
So this is as a fusion protein,
so this is in the context of the full-length virus, right? So replication competent, not just a expression of an individual viral protein.
Okay. So that whereas the GFP is in the particle, or is it attached to protein?
It's attached to the protein. So it's in the locus, within. So these are bacterial artificial chromosomes, let's say BAC viruses, where you can engineer the position of the green fluorescent protein to be
anywhere you want.
Yes.
So then in the cell, when the virus is infecting and producing proteins, how does GFP fit into the mass spec part of it? So
the GFP fits into your ability to visualize when that viral protein when is it produced and where it is localized, and it provides you with a great hook for immunoaffinity purification of that viral protein at different time points of infection to find out all these sort of intricate protein complexes that are formed between viruses and the host proteins.
Got it.
Right? And this is what triggered our interest in my lab's interest in chromatin remodeling enzymes. But this is how we got interested in this protein called interferon-inducible protein IFI16. Because we found it during infection with human cytomegalovirus, we found it as being targeted by a tegument protein, so protein present in the virion, infectious virion, and the most abundant tegument protein in HCMV. It's called UL83.
Now, how did you find that interaction? By mass spec?
By mass spectrometry.
I shouldn't ask that.
Yes. And I was surprised. So
was that of the virus particle or infected
no,
cell?
No, that was from infected
right.
primary human fibroblasts that were infected with human cytomegalovirus, and we found that this tegument protein goes quickly to the nucleus and immediately interacts with IFI16, with this interferon-inducible protein 16.
Got it.
And this is how we actually became interested in this protein and said, why did this very abundant viral protein acquire this function to immediately go and bind, and this was the most prominent interaction of this viral protein in the cell. And this is how I became really interested in this topic that I think it's of so much interest these days, and I think it's a great time to be in this field of DNA sensing because there are lots of things happening and many really remarkable directions that are opening. And it has to do with this fundamental question of how our cells, how our mammalian cells are able to recognize viral DNA versus our own human DNA.
Yeah, it's DNA, right?
It's DNA, it's the same molecule. And as you pointed out, I think that's what led to this long-standing belief that maybe the way this happens is by having this sensing of viral DNA in a subcellular compartment that doesn't contain normally our own human DNA.
Correct.
Right. And that's why the sensors that have been identified so far have been cytoplasmic or endosomal, and it's only been recently that this concept of nuclear sensing of viral DNA has emerged. And
so how did you find that IFI16 was a sensor?
So we found that IFI16 can bind to viral DNA through one of its domains, through these heme domains, and it finds really effectively and in a sequence-independent manner, right? And we found actually another protein in the same family with IFI16. It's called IFI-X, interferon-inducible protein X, that also can bind through a similar domain, these heme domains. So usually people, when they refer to sensors, these proteins have to fulfill two requirements. They have to be able to bind, recognize and bind to viral DNA, and they have to be able to induce the expression of antiviral cytokines or immune response in following that recognition of viral DNA. And we found that IFI16 is also needed, required for this induction of interferon response upon infection.
And so how does it do that? It binds DNA, and what are the subsequent steps?
So it binds DNA, and it seems to bind DNA in the nucleus, and we showed this by fluorescence in situ hybridization, where we showed that that's where it binds to viral DNA. And then it seems to trigger interferon response, but still by requiring the adapter protein STING in the ER. Right? So how exactly the immune signal is propagated from the nucleus, from IFI16 to STING, and that's still not very well understood. But a downstream pathway has been very well characterized in the past even from the cytoplasmic DNA sensors that also signal through STING. And it's through
C-gas is one of those?
C-gas is one of them. James Chen has really put tremendous effort in identifying, characterizing C-gas as an important DNA sensor in the cytoplasm.
So STING is a intermediate like MAVs would be on the mitochondria for RNA sensing.
Yes, exactly.
And it's part of a pathway, and the end result is
the induction,
transcription.
Transcription, and yes, exactly.
But STING resides in the ER.
Yes.
So does IFI-16 go there and interact with STING?
So we do not have for the moment any evidence for that, right? What we do know, however, this is something that we are still working on, we find that if we infect the cells with viruses that are not having the ability to inhibit IFI-16, so for example, during infection with human cytomegalovirus, if we use a strain that lacks the expression of that tegumen protein that I mentioned earlier, UL83, then this virus suddenly, it's an viral infection, can replicate, but it can no longer inhibit IFI-16. And this virus triggers much higher induction of antiviral cytokines because our mammalian cells can trigger that. Well, if we infect these cells with these viruses, we see that IFI-16 tends to go to puncta within the nucleus and tends to oligomerize through one of its domains at the N-terminus. It's called domain called pyrene domain, and it oligomerizes, and it seems to go to these PML nuclear bodies, also called ND10 bodies, and that's where it starts to oligomerize. So,
and that's a prerequisite for STING signaling?
It may be.
Maybe.
This is something that we are still working on.
Okay.
But I think it brings into play quite a few interesting functions of IFI-16 as a DNA sensor, but also as a transcription factor and a regulator of viral gene expression.
These PML bodies are quite complicated, all the things they do,
yes.
right?
And it's very well known that the genome of herpes virus is deposited either in proximity to PMLs or at PMLs, and that most viruses have acquired these mechanisms for suppressing these PML functions and their antiviral and defense responses, right?
So you also found that a herpes simplex
yes.
protein had the similar function and interacts with IFI-16?
So during infection with herpes simplex virus 1, HSV1, we found that the viral E3 ubiquitin ligase, ICP0, is involved in targeting IFI-16 very quickly for degradation. And that happens very quickly upon infection. So here you have two related viruses, an alpha herpes virus and a beta herpes virus, that have found different solutions to the same problem. One of them quickly targets IFI-16 for degradation, and the other one says, no, I'm going to actually hijack this. I'm just going to block its ability to oligomerize. The tegumen protein that I mentioned, UL83, binds to the pyrene domains and stops it from oligomerizing. So it stays there in the nucleus. It's brilliant. Intriguing because IFI-16 still retains its ability to bind to viral DNA, but then it seems to be used probably as a transcription factor or something else in terms of how it regulates viral gene expression.
But not for herpes simplex, which simply degrades it, right?
Yes.
And what's important here is that for both of these viruses, it's a virus particle protein that's doing this, which makes sense because that way you don't need viral gene expression to make the protein. Right.
So for HSV1, actually, it seems that you do need viral gene expression but it's the immediate early viral gene that it's. So ICP0 is present in the virion, but it's
not enough.
It may not be enough, but it's an immediate early protein. So there is still debate in the field if ICP0 alone does this, or if there are other viral proteins that are helping it as well.
So you also find that...
And David Knight at Harvard also, he really put significant work in understanding how IFI-16 is regulated during HSV1 infection. So
you also found that IFI-16 is not always nuclear, right?
Yes. So IFI-16, depending on the cell type, can really have different subcellular localization. So in a fibroblast or in a HEC293 cells, we see it predominantly nuclear or almost exclusively nuclear.
So it has a nuclear import?
It has a nuclear localization signal, a multipartite nuclear localization signal that we defined a few years ago. And we found that if it gets acetylated within this nuclear localization signal, then its import into the nucleus is inhibited, is blocked.
So the acetylation occurs on the nuclear?
On the nuclear localization signals.
On a lysine?
On a lysine, yes. Two lysines in two different NLS motifs.
And you figured that out by
mass spectrometry?
Right, by looking at cells with the protein in the cytoplasm versus the nucleus?
Yes, by fractionating the cells and looking at the protein, and also just by enriching it, by immunoaffinity purifying it and characterizing all its post-translation modifications.
So what causes it to be acetylated?
So we found that P300 is the acetyltransferase responsible for acetylating one of the motifs, and we think that some of the cytoplasmic HDACs, so far our evidence points that way for deacetylating these residues. It seems like the newly synthesized proteins is the one that is acetylated and no longer can go into the nucleus upon certain stimuli.
And what do you think is the role of having cytoplasmic
So
IFI?
it would be extraordinary if the same sensor can be, it's a two-signal model that would use one molecule for multiple function and to detect the different pathogens in different subcellular compartments. But coming back even to the idea that it may not be only acetylation. Acetylation obviously regulates the protein localization, but in different cell types, THP1s, monocytes, the distribution of IFI16 in the nucleus and the cytoplasm differs. There is much more cytoplasmic IFI16 than in fibroblasts that hardly have any. Right? So I don't think that there is enough knowledge out there yet regarding the localization of IFI16 throughout our body in all these different cells that can respond to viral infection. And where would it be, if we step back and we look at the bigger picture, when would it be relevant for IFI16 to be in the nucleus versus cytoplasm?
Yeah, I'm trying to think of situations other than a pox virus infected cell where there would be cytoplasmic DNA.
Because as you know, the majority of DNA viruses that we know of replicate in the nucleus.
Right.
But there is also this thinking that in certain cell types there can be some disruption caused to the capsid that may trigger some leakage.
Yeah, I just find that...
We don't see that in fibroblasts. We really do not.
Yeah.
So this simple identification of an NLS was so tremendous for us because we could then target the localization of the protein either to the nucleus or the cytoplasm and ask where does it sense this viral DNA during an HSV1 infection or during HCMV infection. And we found that in the nucleus is where it binds to viral DNA and triggers interferon response.
So you have an IFI16 that will not ever be cytoplasmic, right? It cannot be acetylated.
Yes.
So if you take cells that produce that and infect them with a pox virus that makes...
We haven't done that experiment, but that's a great experiment to do.
It'd be interesting to know if there's any... I don't know the literature well enough to know if there's a big innate response to pox infections, whether the virus certainly encodes antagonists, all these PKR antagonists and so forth, right?
Right. So that's really an important point because we may be studying viral infection in a condition where that virus already found the problem, solution. So therefore, you're studying something that is already inhibited, right?
So you just take it away. There are certainly virus mutants lacking these antagonists. Yes. You could infect your cells. It would be interesting to know what these cytoplasmic sensors do. I mean, the CGASH system is incredibly beautiful, right?
Yes.
But what is it doing in the cytoplasm? But in your case, you've shown it collaborates with at least the STING part of it is collaborating with that.
And David Knipe at Harvard also showed that CGAS in fibroblast, in human fibroblast, upon infection with HSV1, it seems to go also to the nucleus. And we saw it in our system by using mass spectrometry that actually it interacts with IFI16.
Yeah.
So that's still an area that needs much more investigation.
Are people thinking that maybe certain intracellular bacterial pathogens might be sensed by these cytoplasmic sensors?
Yes, and there has been work already done in that with CGAS, right?
What about the idea that sometimes nuclear DNA leaks into the cytoplasm, right? And then you have to take care of that because that's not good. I don't know why it wouldn't be good, but does that make any sense? Then you turn on interferon, but...
It makes sense, and if we think about an infection and the drastic changes that it causes, morphology changes that it causes in a cell. And if we take HCMV, it generates this large nucleus. Obviously, there are things that could leak. That would be, however, a bit later in infection, while this first immune response an interferon response should be very quick, right, early on. But later in infection, I would not be surprised to find out more and more examples when nuclear DNA, or we saw recently those really interesting papers about mitochondrial DNA also leaking and triggering immune response from the cytoplasm.
Now, during mitosis, right, the nuclear membrane is gone and DNA is everywhere, right?
Yes, right.
So
then
what happens then
if they are silenced, or if they are if there is expression, or maybe the turnover, how is regulated, that we don't know.
It must be regulated in some way, otherwise you'd be making interferon left and right during mitosis, right?
Right, yes.
Well, though protein synthesis is inhibited in general, right, during mitosis, so that may be part of it. You just turn everything off, let the DNA be in the cytoplasm for a while, and then you take care of that. A lot of interesting questions left,
so many interesting questions left, and from our interest on chromatin remodeling enzymes and histone deacetylases, we looked at these sort of questions during cell cycle progression and found that a simple phosphorylation event by Aurora B on these HDACs immediately triggers a completely different localization to the spindle midzone, and suddenly they are away from the reforming nuclei and their transcription repressive functions. We do not know yet if something similar is for IFI16, if there are some modifications that recruit it somewhere, or if it's just simply its synthesis that it's stalled.
So one thing I noticed in your published work is that there's some kind of interaction between IFI16 and C-gas.
Yes.
What's that?
So we found it in fibroblasts, and we found it in THP1 cells. Whenever we isolated IFI16, we also found C-gas there to interact with. And this is what triggered our interest in finding out, is this a nuclear-cytoplasmic interaction. And I can't say that we have a completely definitive answer, but what we know so far is during HSV1 infection, it seems that a subset of C-gas seems to go to the nucleus and interact with IFI16, and we had all these results in hand, and I met David Knipe at one of the conferences, and we were talking about this, and it turns out he was very interested in this same topic, and they were investigating C-gas, and this is kind of how we started the collaboration, and his lab showed that C-gas modulates the stability of IFI16, but that was all his work.
Okay, and so we don't know what the meaning of this is yet.
No, I don't think we know.
But it sounds like there is some interaction between the nuclear and cytoplasmic sensing machinery.
Yes.
Which would make sense.
Right.
And what that is and what it means remains to be determined.
And we find other interferon-inducible proteins that are known to interact with STING, that upon infection they seem to go very quickly to the nucleus and interact with IFI16. So these proteins would have, of course, the ability to also shuttle back and activate STING. We are now using live cell imaging and microscopy to really put all these events in a proper temporal nature.
Okay, so I think we've talked well about IFI16
and DNA sensing.
And DNA sensing. Let's turn to something totally different, which I heard you talk about at ASV, in which you've also written about Sirtuins and virus infections. What's that about? So
this was connected to my interest when I started the lab, right? This
chromatin remodeling.
Chromatin remodeling and histone deacetylases.
Okay.
So what we found is that, as I mentioned, viruses that are so different from each other, and they target these enzymes, and very prominent in our isolations and our studies of virus-host protein interactions, were all these histone deacetylases, also called HDACs. But also Sirtuins. Sirtuins are class 3 HDACs, are NAD plus dependent deacetylases, so they're enzymes that remove acetyl groups from lysine residues on proteins.
And what does that do?
So usually that's connected. So for Sirtuins, these are really interesting enzymes. Some of them are nuclear, one is cytoplasmic, many are mitochondrial. For HDACs, again, they are nuclear and some of them are cytoplasmic. For the nuclear ones that were the best study for both Sirtuins and HDACs, is this sort of removal of acetyl group from histone proteins is thought to be connected with transcriptional repression. Right.
It tightens the chromatin.
Tightens the chromatin, and yes, and so these are considered epigenetic factors like Sirtuin 1, that is nuclear and it's very well studied, and in its regulation of histone 3.
And these have global effects, right?
Global effects on the cell and on gene expression. However, there are these other Sirtuins, as I mentioned, that are mitochondrial, that are also removing acetyl groups there,
in the
Sirtuin 3, in the mitochondria,
DNA.
on mitochondrial proteins, and it seems that there are so many.
But not histones.
Not histones.
No histones in the mitochondria,
yes.
Right, okay.
There are so many mitochondrial enzymes that seem to be regulated by acetylation.
Has it been known for a long time?
It's fairly recent, just recent years that it starts to be understood that acetylation is a really prominent post-translation modification in the cell. So it was not as studied, but in the last 5-7 years, more and more studies of acetylation have come up, and now it's clear that there are thousands of proteins that are acetylated in our cells. And
besides histones.
Besides histones.
Maybe that was, people were so focused on histones, they didn't look elsewhere.
It's, I think HDAC also as a histone deacetylase is a wrong terminology, because they modulate the acetylation status of so many other proteins. Okay. Tubulin, P53, right? Really important proteins in our cells that are regulated by acetylation.
Are you saying that it's not all about phosphorylation?
No, it's not all about phosphorylation. So, so given the fact that we saw all these Sirtuins targeted by viral infection, we wondered why, and wanted to understand a bit
targeted
better.
Meaning degraded, or?
Meaning that they were interacting with, when we isolated different viral proteins, we found that in the interaction, yes.
By mass spec?
By mass spectrometry. And all this post-translation also by mass spectrometry.
Okay. These are herpes viruses, right?
These are herpes viruses, but also with HIV we found the same. And actually before our work on Sirtuins, most of the studies done on Sirtuins and viral infections have been with HIV and Sirtuin 1, this nuclear protein that it's an epigenetic factor that modulates histone acetylation, and it was shown that Sirtuin 1 can regulate the acetylation level of an HIV TAT protein.
Has it been looked at for any other RNA viruses besides retroviruses? Do you know?
So not enough. So there haven't been too many studies on Sirtuins with viral infection. I think now, in these recent years, there started to be a few, but in the past they
Just wondering how broadly this modulation of Sirtuins is, right?
Right. So what we started to look at it, and I went immediately to talk to Tom Schenk across the corridor here.
He had been interested in this already, is that correct? Or was it your...
So I don't know if he was interested. I don't think he worked on Sirtuins before. We were working already on Sirtuins and just trying to understand also what they do outside of the context of infection, because some of them are really poorly characterized. Sirtuin 7, Sirtuin 6, and when we started there were hardly any papers on this. So when I went to Tom and asked him, let's do this, it was because he had this beautiful system set up for knocking down individual genes and looking at the impact on virus titers. And he was doing this with the kinome, kind of looking at the kinases. So I said we could just add, the Sirtuins to the screen, and he was very receptive and immediately was interested in this. And this is how we noticed that if we knock down any one of the 7 human Sirtuins, the viral titers were significantly increased. And what was surprising to us that this was a broad phenomenon for most viruses that we tested. So for human cytomegalovirus, for herpes simplex virus 1, for adenovirus, so several DNA viruses, but also for influenza A. You were asking about another RNA virus. Again, the same
and
issue.
all of them, all the Sirtuins have knocking down all of them has this effect?
Knocking down each one of them individually triggers increases. However, there are slight differences. So for example, in influenza A infection, it seems the mitochondrial ones do not have as such a striking impact on viral titers, maybe because of the requirements of influenza of H1N1 for different requirements than an HCMV or herpes virus on cellular metabolism, right? We know that viral infection triggers all these striking changes in cellular metabolism.
Where did the name Sirtuin come from, by the way? Do you know?
From the name of the protein in yeast, Sirtu, that's the homologue.
SIR2,
yes, SIR2.
Okay.
But what was striking is that it seems like this antiviral or defense property is really broad, and it's also evolutionarily conserved. Hanna Budayeva in my lab, a PhD student, showed that in E. coli, COB-B, that's the Sirtuin homologue, when that one, that also has defense properties against many different bacteriophages. So we are looking at a defense mechanism that it's quite old and fundamental to cells.
So the fact that it's in bacteria means it's not involving the known eukaryotic innate or defenses, for example.
Yes.
It's not an interferon-based defense.
It doesn't seem to be, although some of the Sirtuins are known to regulate immune responses. Sirtuin 6, for example, regulates NF-kappa-B as well. And so we started then to be interested in understanding how does this work, all these mechanisms that are out there. Is there a unifying theme and one mechanism, or are we looking at many different mechanisms here? And one of the Sirtuins that gave the highest impact on virus titers was Sirtuin 4. That's a mitochondrial Sirtuin. And also it has the highest homology with bacteria, the Sirtuins. So Sirtuin 4 is an interesting one because in the mitochondria we know of 3 Sirtuins. Sirtuin 3, that's a really strong deacetylase. It can remove very effectively acetylation modifications from proteins. Sirtuin 5 was recently described by several labs, including Yingming Zhao at University of Chicago, as a demalonylase and desuccinylase. So it removes malonyl groups and succinyl groups from lysine residues.
Okay.
But Sirtuin 4 didn't quite have a very strong enzymatic activity. It was known as an ADP ribosyl transferase activity, quite a poor deacetylation activity. So we wonder how comes, what gives it this intriguing...
Well, by the way, where is it in the mitochondria?
So it's thought to be in the matrix.
The part that's named after you. Right?
Yes. And but
the matrix, okay.
Yes, but Sirtuin 4 is so low levels, so such low abundance, that it hasn't been enough characterized, and I don't think it's. I think we will discover probably much more about this enzyme in recent years. So we purified mitochondria, and we looked in the mitochondria what this protein, what this enzyme interacts with, to find out what it's up to. And we found that it interacts with these protein complexes, dehydrogenase protein complexes, like the pyruvate dehydrogenase complex. And what these complexes in the mitochondria had in common was that they had this really peculiar modification on them, lipoylation.
The addition of lipid.
Yes, the addition of a lipoyl group. Lipoyl group.
Okay.
Yes, not, and.
I'm going to see what a lipoyl.
Oh, I should show it to you. I have it.
Oh, here it is.
There it is.
Is that right?
Yes, that's it.
This thing here.
That's the guy.
With sulfurs?
Yes.
It's not really a lipid, that.
No,
right?
it's not a lipid. That's
okay.
So this one.
So you want to go there?
No, it's okay. So that's present on several dehydrogenase complexes in the mitochondria. There you go.
This one here?
Yes.
What's ACP?
I don't know what. Then maybe a label. This is here.
That's
this
the
one.
lipo.
That's the lipo. This is what you're looking at. Yeah.
So it has a ring at one end.
Yes.
With 2 sulfurs.
2 sulfur groups.
And then a carbon chain with and oxygen at the other end.
Yes.
So this is attached to proteins
Two lysine residues on this
by
protein.
Sirtuin 4.
It's removed by
Removed
Sirtuin
by
4.
it, okay.
So what
What does this do, this lipoyl? Does it have a function?
Right, yes. So we found that it regulates the activity of the pyruvate dehydrogenase complex, and that Sirtuin 4, by removing this lipoyl group, it inhibits the activity of the pyruvate dehydrogenase. So this is a really important protein complex in the mitochondria. It converts pyruvate to acetyl-CoA. So it links glucose to the TCA cycle, but also to fatty acid synthesis. So
it's part of energy generation, right?
Exactly, yes.
So if you take off the lipoyl group, you inhibit the activity of the enzyme?
You inhibit the activity of the pyruvate dehydrogenase complex,
so
and you decrease the levels of acetyl-CoA, you decrease the level of fatty acids in the cell, which viruses like HCMV really rely on and they need it. And usually they increase the pyruvate dehydrogenase activity, they increase the fatty acid levels.
So the Sirtuins, the Sirtuin 4 removes this.
Yes.
So that's why when you take away Sirtuin 4, HCMV replication goes up because it's getting more lipids, fatty acids, which is what it needs to make its membrane and so forth. Okay.
And we, yes, exactly. And this was really intriguing. So this was the first time that lipoylation was found by mass spectrometry in a cell. It's really hard to detect. These peptides are very badly behaved, even in solution. It's hard to keep them soluble. And we were able to show that in knockout mice, for example, Sirtuin 4 knockout mice have higher PDH activity levels because they do not have Sirtuin 4 there.
And they're fine, these mice?
So they are having quite a few issues, but they seem quite slim.
Okay.
But we are now starting to investigate them to see what actually.
Oh, so you're going to patent this for weight control?
No. Who knows what else it triggers?
But the mice are viable. They live normal lives and so forth.
Yeah. Well, they won't live for too long.
No.
But yes. So most of the Sirtuins, not necessarily Sirtuin 4, but most of the Sirtuin work has been in aging. Because one observation is if you knock out Sirtuins, then the mice die younger, right?
Yeah, someone has made a company based on that, right?
Sirtuins, and
yeah, Sirtuins.
And I think they have even some
Lenny
creams for face if you want. Yes,
right?
Lenny Guarantee, yes.
Oh, that's right. Yeah, I remember that. Does it work?
The cream? I don't know. I didn't try it.
Interesting.
One of my PhD students told me actually about that, Daniel Riles.
I heard a podcast about that by a friend of mine who does science podcasts, and he said, Guarantee says, you could add 20 to 30 years to your life eventually with, I don't know what you're going to do.
Well, so you know that resveratrol is an activator of Sirtuins and is a compound of red wine. So you may want to.
I'm very happy to hear that.
There's another reason why it's good to drink red wine. Although you may need to kill your liver by the time you reach the right resveratrol level.
Well,
but you can buy them in pills, resveratrol.
Well, red wine is more fun, right? All right. So Sirtuins remove.
So Sirtuin 4 is the first known cellular human cellular lipoamidase enzyme, so that removes these lipoyl groups from several dehydrogenase complexes in the mitochondria, including the pyruvate dehydrogenase complex. And so far we just characterized the function. What's the impact of removing this modification from the complex? And we see that it inhibits it, so thereby inhibiting the production of acetyl-CoA and fatty acids.
So so for viruses that need fatty acids, that would explain that, right, as an antiviral. But what about what's the impact on ATP? And viruses need that, obviously, right?
So we haven't done yet a characterization of all changes in the cellular metabolism after, let's say, in Sirtuin 4 knockout mice were overexpressing Sirtuin 4, and that's something we want to do. But one thing to mention is that in our isolations and in our mass spectrometry analysis, we do find that there are other dehydrogenase complexes that are associated with Sirtuin 4 and are also modified by lipoylation. So like the one regulating alpha-ketoglutarate levels, and so there would be other pathways that probably would be regulated through this enzymatic.
It's never as simple as you think, right?
No.
But Sirtuin 4 is exclusively mitochondrial, right?
Yes.
And the others are in different places.
Sirtuin 3 is also mitochondria. So Sirtuin 5 is mitochondrial, but also seems to have another isoform that is cytoplasmic.
And do they have the same activities as 4?
No, so none.
Different.
The other ones are not lipoamidases. Yes,
so that's interesting because they're all antiviral, and they have different mechanisms.
Yes.
So it'd be interesting to tease those out too.
So
right?
of course, one kind of overarching theme is that they are NAD plus dependent enzymes. So, and viruses seem to modulate the NAD plus levels in the cell viral infections, right? And of course, if NAD plus is reduced during an infection, then these enzymes will be inhibited. And it seems that that's what happens during infection with herpes simplex virus 1. The NAD plus levels are reduced. So when we knock down sirtuins, we do see an elevation in viral titers, but not as high as for human cytomegalovirus. It's much higher, and NAD plus levels seem to be constant.
So
there.
do you know how that's achieved by the virus infection?
Not yet, no.
That's interesting, right?
Yes.
Must be a viral protein that's somehow interacting with. So what's?
And I'm wondering how quick that is. It could be something that happens immediately as they enter the cell periphery, and we have done some of those studies. But
so what produces NAD in the cell?
Well, let's see.
You can look it up. It's NAD that's affected by herpes infection. Is that right, or NADPH?
NAD plus, so my lab hasn't done any of those studies. Josh Rabinowitz at Rockefeller and Tom
Who used
Sheng
to be with Tom,
have
right?
done, yes.
They collaborated when...
Yes. So Josh Rabinowitz is a professor in chemistry department at Princeton. No, here.
Oh, he's here.
Yes. And
that's
he's,
right. And he's the one who's got Tom on the whole metabolomic
exactly. So
track,
they are
right?
the ones who have noticed that NAD levels are decreased during HSV1.
So we had Tom on TWiV some time ago.
Yes, yes.
And he said, Rabinowitz said, I have this technology, what can we do with it? And Tom said, well, let's look at infected cells, see what happens to the metabolome, and that's the beginning of that.
And I think now they have started, they are doing beautiful work, but they are comparing the metabolomes after all these different viral infections to trying to figure out what are the changes and how much of does the viral infection require these certain changes.
Well, that's clearly going to intersect with your Sirtuin interest,
yes,
right?
Exactly. So that's why it was such a clear match when I went to talk to Tom about Sirtuins.
Right.
And
interesting. So what is it that you want to figure out about? Do you want to look at all these Sirtuins, or are you going to focus on 4?
So I do want to understand a bit better what 4 is doing, and because of its impact on herpes viruses and really and all DNA viruses, when we knock down Sirtuin 4, it triggered the highest increases in virus titers. But we are having projects in the lab also working on the other Sirtuins. Elizabeth Rowland in my lab works on Sirtuin 4, but Shinlei works on Sirtuin 3, finding out how this deacetylase enzyme works. And Hanna Budayeva works on Sirtuin 1 and 2, and she's very interested in how they regulate P53 during infection.
This is all in the context of herpes virus infections,
This is all in the context of HCMV and HSV1 infections.
So your goal is to understand how the Sirtuins and the removal of lipoyl groups, how that impacts virus replication, right? And,
yes.
And probably in doing that, you'll understand the normal pathways in uninfected cells as well. Right?
Yes, and it gets even, it's never that simple, right? It always gets a bit difficult. So for example, Sirtuin 4, later in infection, Elizabeth in my lab showed that it seems like it's probably inhibited by a viral protein.
Its activity is inhibited?
Yes. So we are now trying to figure out that mechanism and find out what's this sort of...
Makes sense, right?
Yes. Well, Sirtuin 3 doesn't seem to be inhibited.
Right. But the virus would want to inhibit 4 because 4 is inhibiting the virus by removing lipoyl groups, right? It's fascinating.
Yes, so we would want to activate it. We would want to find a way to activate Sirtuin-4.
This is great. So this is a whole new area, right?
Yes, and Todd Greco, a postdoc in my lab, and Rommel Matthias, who was a postdoc in my lab and worked on Sirtuin-4 and now moved to Australia to start his lab. But they really had to start from scratch all this methodology for studying lipoylation and understanding Sirtuin-4, and it has been fantastic.
So I remember at ASV you were very excited to talk about the Sirtuin.
Yes.
Is this going to be the main focus going forward of your lab?
Well, I think if I would say that, half of my lab would throw their hands in the air and leave me, because half of the lab continues really on DNA sensing. We just can't get enough of that. It's just
that's good.
We think that it's so important, and it's still too many questions out there, and we think we can help with that.
How big is your lab?
Right now it's not too big. It's about 8 people.
That's pretty good.
And I like this sort of size when I can still speak to the people in my lab, and I find that sometimes these small labs are producing just as much as larger labs.
There's this whole idea that big labs are not very efficient, right?
Yes.
NIH supports a lot of big labs, but they get a lot of money. It's not clear that it's a good return on the investment.
Right. I think it's probably depending on case to case, but if I look at many large labs, I don't see them producing more papers than we are.
You're amazingly productive. I've looked at your CV.
I have an amazing group.
So many papers. But one thing I wanted to ask you, you also collaborate a lot with other people. And how does that work? Is that, I mean, you have to watch out and not do it too much.
Yes, exactly, and
right?
not to spread too thin. But in the same time, collaborations can be great for anyone in my lab, because it makes you think about a different project from a different perspective, and usually there are projects that are somehow connected to what they work on, right? So every person in my lab would have their own project, but then they will have one or two couple of small collaborations that do not take too much of their time, but they are designed to make them talk to different people and interact, and they will get access to different approaches, and it works really
and maybe you
beautifully.
can develop something new that you can take with you at some point if you're a postdoc, right?
Right.
So you don't work in the lab anymore, I presume?
No, unfortunately not that much. And this is, I used to in the beginning, I can't believe it. I remember when I started my lab, and I would be petrified to go to a conference, because I would get anyway a call to ask me, where is that antibody? It's in the third drawer. Those times are gone. And that's because I have such good people in the lab, and they can train each other as well. But this is why I absolutely love teaching this summer course at Cold Spring Harbor Laboratory. I am an instructor for the summer proteomics course, and that's because I'm in the lab and doing all the experiments with my own 2 hands and showing people, and I think it's entertaining also for the people in my lab to say, yes, she can actually do all this.
So this is a course where you teach people to do mass spec, is that right?
Yes, so we actually teach a lot of things. We teach how to study protein complexes, how to study phosphorylation, so you see it's still important.
Yeah, of course.
And how to do targeted quantification, something we call selective reaction monitoring, how to do that. And we put a lot of emphasis on data interpretation and analysis. That's really so important these days. And of course, we teach the fundamentals of mass spectrometry, but a lot of it is really front end and then back end, how you analyze.
So you get to be in the lab and doing things.
Yes.
How many weeks is this?
Well, when I've been teaching this for 11 years, and when I started it was 3 weeks, and now it's 2 weeks, because it's
that's too bad.
2 weeks is a bit.
It's a long time, yeah.
It's more manageable.
That's great. And
we install mass spectrometers for every year for the course, just before the course.
Oh, it's just for the course, and then you take them out.
Yes.
Wow.
So it's a lot of work.
So do you also teach a course here at Princeton?
Yes. So I teach a core course that it's cellular biochemistry, and it's a core course for the graduate students, and that's in the fall. And I really love that, because I get to know the graduate students very early on. And I teach in the spring a small module on proteomics in biological sciences in the main introductory course, the basic introductory course for undergraduate students in molecular biology.
Okay.
And next year I'm starting actually to teach biochemistry for the undergrads. In
So
the spring.
these are bigger courses.
These will be bigger courses.
That's a course that Jane Flint used to teach,
yes,
right?
Yes, right.
She always complained about it.
And Fred Houston now is teaching this course in the fall and is really going so well.
Right.
It's
Wonderful. I started teaching undergrads at Columbia 8 years ago. I mean, I'm at the medical
Yes.
school, but I don't have to teach undergrads. But I love it because everything is exciting and
It's
new.
fresh, isn't it? Yes.
It's really a lot of fun. I also noticed that you are a senior editor of this new journal, M Systems. That
Yes,
ASM
right. Yes.
is. What is that going to be about?
So it's going to give another flavor. It's about systems biology, right? And introducing all these proteomic, genomic, microbiome, sort of larger studies, but they are really in the microbiology, applied to the microbiology field, and we do expect really solid biology and mechanism working to a mechanism, but there are not too many opportunities for such publications. There is a really nice journal, Molecular Systems Biology, but outside of this, unless you publish a cell reports paper, resource paper,
Right.
not too many. And these days, we all generate these larger data sets that are really informative and takes great skill to analyze and interpret that. And so this is going to be a great avenue.
Then that's starting in January, I
Yes,
think, right?
it's going to start next year.
Well, this should be exciting, right?
Yes, very exciting.
A new journal, you can sort of steer it the way you want, right?
Yes.
It's great.
And I'm also on the editorial board for Molecular Cellular Proteomics, and that's the top journal in the proteomics field, and they really put a lot of emphasis on biology and really finding something important.
Wonderful. Well, I don't know where you get the time to do all this. You're incredibly productive. You do all these things.
I love it. I feel that somebody pays me to do my hobby, for my hobbies, which I think is a great thing.
That's how I feel when people ask me. I say, if you love something, it's not work, right?
Yes, right.
And it's great. Is there anything we missed that we should have touched on?
Yes, of course, many things. I think you have to come here more often. And
I would love to, absolutely. I can come here once a year and we can catch up on what's new.
Yes.
I'd be happy to do that. Well, this episode of TWiV, just like all the others, will be found at twiv.tv, also on iTunes. You can get it on your iOS or Android device. There are lots of podcatching apps that you can download for free and subscribe for free. And if you have any questions or comments, please send them to twiv@twiv.tv. I want to thank my guest today here at Princeton University, Ileana Cristea. Thank you so much for
Thank you for having me. This has been wonderful.
It wasn't so bad, right?
No, not too bad. If I said something inappropriate, you'll cut it off later,
I don't think you
right?
said anything inappropriate, not at all. I think it was all great, and I really appreciate your taking the time. I love your work. I think you're fabulous, and you must love being here, right? This is a dream place to be. So
it's an extraordinary department.
It's just, there's so many good people. We'll put a link to the department. You'll see all the awesome people who are here, and many of whom I know, of course.
And a great, if I may add, a great graduate program.
Yeah.
Okay, sure. I'm a little bit biased. I'm the chair of graduate admission,
great.
but it's a fantastic graduate program. It's so multidisciplinary and so diverse. It's just an incredibly rich scientific environment here.
So molecular biology, which is this department, has its own graduate program?
Yes, its own graduate program, and it also is very interactive. So our students can do rotations in Louis Sigler, in the genomics, in chemistry, in all these different programs.
How many faculty
physics.
are there in molecular biology?
Well, depends how we count them, because if they're also the affiliated ones or not, but we are a fairly large department. It goes up to 50 something.
Must be some faculty meetings then.
Yes. Our faculty meetings are fantastic because we have every Tuesday we have lunch together, and one of us presents research from their lab.
Very good.
And it's a really nice discussion, scientific discussion.
50 faculty, it'll take you a year, and then you start over again.
Yes.
And your chair is Bonnie Bassler.
Bonnie Bassler.
And before that, it was Lynn Enquist.
Yes.
and I think Tom Schenk.
Tom Schenk. We had,
Arnie Levine before
yes.
that, and that's as far back as I go.
We had quite a few virologists.
You had a lot of virologists.
And microbiologists
right.
now with Bonnie.
The funny thing is, Levine, Schenk, and Enquist were all editors-in-chief of Journal of Virology.
Ah, okay.
Princeton Journal of Virology connection here.
Yes.
That's funny. I'm Vincent Racaniello. You can find me at virology.ws. You've been listening to This Week in Virology. Thanks for joining us. We'll be back next week. Another TWiV is viral.
By Thermo. So this is one of the mass spectrometers in my lab. We have a liquid chromatography unit here, and then the samples enter through this into the mass spectrometer. This is an electrospray ionization, which means the samples are introduced in liquids, and you can see a droplet there. So this is not scanning right now, and we have a monitor here that shows that exact. So if this would be on, you would see a very diffuse spray here and not a liquid.
So that's the actual drop right there?
That's the actual drop, right? And then this is the mass spectrometer. So as I said, it has several different mass analyzers. So in this front part, it has a quadrupole that acts as a filter, and then it has an ion trap that traps the ion. And then here it has an Orbi trap. So that's the has higher accuracy than the ion trap. The ion trap has very good sensitivity, and this one traps the ions and then sends them to the detector. And all these are kind of working non-stop, day and night. They do not need supervision, and they can be controlled also remotely. And then here we have another instrument. This is also an Orbi trap, an LTP Orbi trap, but it's a newer model. And again, it's attached online with a liquid chromatography source, the liquid chromatography unit here. And again, it has an ion trap, an Orbi trap, and it has also, in the back here, it has a unit that allows you to fragment peptides in different ways. So how big a peptide can be or how small. This is an electron transfer dissociation unit, an ETD. So this is great for studying post-translational modification because phosphorylations and so on, they are labile, they can be easily dropped, but this one keeps them on. And you can find exactly the position of the site. So that's another instrument. And as you can see or feel, Vincent, they generate a lot of heat and a lot of noise. So we have this panel for noise reduction.